Presuming that you watched the other video about the movement of molecules across biologic membranes, there are a few clarifications or additional pieces of information that I'd like you to consider. First, it's true that small nonpolar molecules, like gases and steroid hormones, as well as some small polar molecules like water, move across biologic membranes by the process of diffusion. Small nonpolar molecules like oxygen and carbon dioxide, as well as steroid hormones, move through membranes by simple diffusion, meaning that they move through the lipid bilayer in the direction that is dictated by their concentrations on either side of the membrane. Perhaps not clear in the previous video is that water does indeed move by diffusion across membranes, but not by simple diffusion. Instead, water moves across biologic membranes by the process of facilitated diffusion, meaning that proteins in the membrane facilitate the diffusion of water molecules. The proteins specific to allowing water movement across biologic membranes are known as aquaporins, and there are multiple types of aquaporins dependent upon which cell they're expressed in. Speaking about protein-facilitated transport, I do want to highlight that we're not only talking about movement of molecules across the plasma membrane, but indeed molecular movement occurs across all biologic membranes, the membrane at the surface of the cell, as well as membranes within the cell. The proteins that facilitate molecular movement come in two flavors or two varieties. The first, shown in panel A, are transporter proteins or carrier proteins. These proteins specifically bind to the molecule that's meant to be transported and depend upon a shape change when that binding and release occurs. The second type, shown in panel B, are protein channels. Channels are effectively what the name suggests, a channel or a pore, if you will, in the membrane. There is association and recognition of the molecule that's meant to be transported, but no specific binding of that molecule, and therefore no required shape change. Both transporter proteins and channel proteins transport specific molecules. In other words, not anything can bind to a transporter. Not anything can move through a channel. Both are saturable, meaning that transport either through a carrier protein or through a channel protein is limited by the rate at which that transport can occur. Obviously, movement through a channel, because it doesn't require specific binding, is going to be faster than movement through a transporter or carrier protein. However, both types of transport proteins are going to work at a maximal rate. There's only so fast that molecules can be moved through either. When we're talking about active transport, I want to remind you that active transport requires energy. The previous video suggested that that energy always occurred in the form of ATP. That's not necessarily true. There are two types of active transport. Primary active transport that does require ATP hydrolysis, but there's also secondary active transport that requires the passive movement of a second molecule for the first molecule to be transported. Each of the proteins shown in this image are moving these yellow square molecules from one side of the membrane to the other. Looking at the gradient, the concentration of yellow square molecules is higher on the top of the membrane than it is on the bottom. So each of these proteins is actively transporting the yellow square molecules from an area of low concentration to an area where the molecule is more highly concentrated. The molecule on the right is a light-driven pump, meaning that the energy from light is allowing the movement of the yellow square against its concentration gradient. This occurs in plant but not animal cells. The previous video focused upon ATP-driven movement as a form of primary active transport, and this indeed is a very common mechanism by which molecules are moved against their concentration gradients in animal cells. The image on the left shows that coupled transport, where the movement of the yellow square molecule against its concentration gradient is occurring because the movement of red square molecules with their concentration gradient is allowing for that movement to occur. This is secondary active transport. Secondary active transport depends upon the coupled transport or co-transport of two molecules. If those molecules are moving in opposite directions, we refer to that coupled transport as antiport. If, on the other hand, the two molecules are moving in the same direction, we describe that as symport. Of course, if only a single molecule is being moved by a transport protein, the term used to describe that is uniport. The previous video also gave two examples, one of primary active transport and the second of secondary active transport. I do want to clarify that the sodium-potassium pump or the sodium-potassium ATPase is an example of primary active transport because ATP hydrolysis is used to move both sodium and potassium against their concentration gradients. As the previous video suggested, 3 sodium molecules will be moved from inside of the cell to outside, and 2 potassium molecules will be moved from outside of the cell to in. The gradients that are indicated on this image show that both molecules are being moved against their concentration gradients. The previous video suggested that this movement is important in keeping the concentrations of these two very important ions appropriate on either side of the membrane. That is, in fact, true. However, the previous video did not clarify that keeping the ion concentrations appropriate on either side of the membrane is really what drives keeping water concentrations, tonicity, appropriate on either side of the membrane. Ion movement will regulate water movement. If we don't have the proper ion placement on either side of the membrane, we won't have the proper water placement on either side of the membrane. So cell volume is maintained by the sodium-potassium pump. The previous video spoke about how important this pump was in nerve cells, and it is true that it is important in nerve cells, but indeed all cells use the sodium-potassium ATPase to move these two ions and therefore regulate water movement across cell membranes. And finally, the sodium-potassium ATPase is the largest single utilizer of ATP in our bodies. As an example of secondary active transport, the previous video described the sodium glucose symporter, although it didn't name it as such. This transport molecule or transport protein moves two molecules, sodium and glucose, together in the same direction. The direction of movement is from the gastrointestinal lumen into the cytoplasm of epithelial cells. Sodium is in high concentration in the GI lumen, and glucose is in low concentration relative to the cytoplasm of those epithelial cells. The passive movement of sodium from the GI lumen drives the active movement of glucose. Both molecules must bind to the luminal side of the transport protein, and when they do, the transport protein changes shape, and that conformational shift allows the movement of both molecules to the cytoplasmic side of the membrane.